All right, hello again. For this video, I want to start getting into talking about this interface where the immune system contacts a pathogen. In this case, I'm going to do this in the context of a virus. So I want to start off just by reading a little excerpt from one of the textbooks that that I've been reading for years now, but this is the main source of the information I'm presenting aside from what I've learned in my classes. It's talking about the immune system, and it says here, just briefly, conveying immunological complexity is challenging. The field of immunology is bewildering to many, even to those who work in closely related disciplines. Contemplating the sheer number and diversity of cell types, soluble proteins, signal transduction pathways, and anatomical locations can be overwhelming. Everything appears to interact with everything else, and there's no evident foundation on which subsequent layers of information can be built. Further, it goes on to say, the immune response is not merely an assortment of different cells with distinct functions, nor does it function in a prescribed sequence of events in which A leads to B, then to C, independently of the pathogen. I wanted to start off with that because just to reiterate the point, immunology can be really confusing because there is a lot going on. And so the coming slides here for this video are really busy slides. I'm not going to go into a lot of the details on them, but I want to convey that message that when people present you some finding, you need to be somewhat scrutinizing that in that to understand what that finding means, you need to sort of understand these underlying pathways, because just because you find something works in maybe a dish in tissue culture doesn't mean it's going to work in a living organism. There's lots of things that happen in lab that we don't see any biological significance in a real organism. And so just carry that thought with you as you watch these videos or as you learn things on your own. The immune system is extremely complex and interactive in its own system. So something to bear in mind. All right, so I wanted to basically start off on how a virus becomes recognized then by the immune system. And I'm sort of skipping over what we would call intrinsic immunity, but these are like the very first immune responses that almost any cell can initiate. And I'm kind of jumping right to sort of the innate immune recognition. I previously mentioned that there are two arms of the immune system we think of, which is the innate immunity and the adaptive immunity. And the innate immunity has some features about it that are very different than the adaptive. And so if you haven't seen that, I made a video on that, and maybe check that out. This is getting more into how the cell actually knows that it has been infected. And so this occurs when the virus makes it past the chemical and physical barriers which were keeping it out in the first place. And now it has found a cell which is permissive and susceptible and permissive to infection. So the way that the cell recognizes that it's encountered a virus is through these proteins up here. And these are just representative proteins of these broad classes. So these are representatives of the toll-like receptors. And these are the C-type lectin receptors, just again, representatives. There's probably hundreds of pattern recognition receptors like these. And so I have that definition down here. Pattern recognition receptors are these protein receptors, which may be glycosylated as well, but they're primarily a protein, and they recognize these things that we call PAMPs, or pathogen-associated molecular patterns. These are unique microbial patterns which our eukaryotic cells do not have. And so that's one way the immune system can differentiate between self and non-self. This is really important because you don't want to attack your own cells unnecessarily, which is what occurs in something like autoimmune disease. So once a pathogen binds, we'll just say a virus binds to one of these, it sends off this alarm signal basically, and it's a cascade of of signaling through the cell, which eventually leads to what we call an immune response. So getting a little more into detail here, I want to show you what these pathways, and again, this is going to be a simplified pathway, but just the complexity involved here. So this is one overview of some signaling that occurs from pattern recognition receptors. And for this image, I want to focus on these boxed ones that I boxed off here. These are what we call the toll-like receptors. They're the prototypic pattern recognition receptor that will pick up on viruses, bacteria, fungi, lots of things. So the way that this works is a virus, which is so orienting you here. This is the cell membrane right here, and everything underneath this is inside of the cell, and everything out here is outside of the cell. And so if a virus is coming along and it binds to a toll-like receptor on the outside here, this toll-like receptor would dimerize, so it would come together, and it would start sending these signals, and it would have an outcome it would have a different outcome depending on which of these toll-like receptors was bound. And each of them seems to really interact with a very specific class of molecules. So for example, TLR5, I believe, is the flagellin toll-like receptor. So this recognizes bacteria. TLR4, I think, is lipopolysaccharide, that it recognizes that, which is a component of bacterial cell walls, or on the outside rather. I think TLR2 is single-stranded RNA, and so maybe that's a good example. I apologize if it's not. I mean, you don't need to memorize these things, but it is kind of helpful sometimes to know what these are. I'm pretty sure this is single-stranded RNA though. And so what would happen then though is, let's say a virus bound to this TLR2, it would send off the signal through these MyD88 proteins, through the help of these MyD88 proteins and this adapter protein here. This comes together, and then it moves that signal down to this IRAC, and then IRAC sends this signal transduction down through TRAF, another adapter protein, and you can see how this gets really convoluted pretty quickly. The details aren't important. What is important though is to recognize a virus bound to an alarm system, essentially, and that alarm system sounded, and through many proteins involved in signal transduction, you wind up with a transcription factor which goes to the nucleus, and it will begin to initiate transcription of a program which will help clear the virus. And these are typically like inflammatory cytokines, or these things that we call type 1 interferons. So type 1 interferons are your classic antiviral response. They consist of interferon alpha, interferon beta. And these cytokines will help mediate the clearance of whatever infection it is. So again, if this seems confusing, one, it is, and really the details aren't important, but the summary of this is that a virus or a microbe will bind to a receptor on the outside. You will have a transduction of some sort of signal, letting the cell know I found something, and it's a virus or it's a bacterium or something, and we need to initiate a gene transcription program that will eventually lead to a protein. And those things should be familiar to you if you've watched the previous videos where I explained this is why you need to know what transcription and translation is. It's because none of this really makes sense unless you understand that. And I put them in very basic terms, so I hope that those make sense. All right, so another confusing slide here. Again, the details aren't important, but what I want you to pay attention to is these red lines with the sort of T, these sideways T's. So viruses have to be a successful pathogen. If a virus were to bind to that receptor on the outside, and it gets internalized and it makes it into the cell, but at the same time, the cell now is sending off all of these signals, and it's saying, hey, we have an invader here, we need to get rid of it. Every single successful pathogenic virus has some way to antagonize or to inhibit these signals. And so that's what these red lines represent. They represent areas where these are the viral proteins like UL37, and this is the virus it comes from. So VP11, here's UL48 on the virus I work on, human cytomegalovirus. And so just as this is an example then, this portion right here, this UL48 protein that human cytomegalovirus produces will stop the signaling of STING. and it won't move from the ER and be able to complete this TBK1 phosphorylation. So that would be right here, sting. So this is where this fits in that bigger pathway. It's antagonizing it right here. And so that basically allows the virus to turn the alarm off. And these are just the various ways that viruses can do that. This is no means like a comprehensive list. There are thousands, hundreds of thousands of ways this occurs in viruses. And so just to give you an appreciation of, how does a virus become pathogenic? It's because it has these sort of counter offenses to the defenses that cells put in place. So as I list here, this can happen any step along this pathway. In this pathway, this can be blocked at every single step, potentially, by a virus. And I've mentioned this, they have to exploit the immune defenses to make sure that they can persist. And one of the things that I think is really cool, and I'll talk about on the next slide, is viruses can prevent apoptosis. And so I'll move on to the next slide and explain it more there. So what is a cell that's been infected and it has the virus, which is trying to stop its immune signaling? Luckily, cells have redundant systems. They have many systems, so even a successful pathogen can't stop them all. So the cell is experiencing this infection and it's trying to alert other cells. And so what occurs? What does the cell actually do from here? It's recognized the pathogen. It recognizes the pathogen is trying to shut it down. And so what is its response? I mentioned apoptosis is one. This is a programmed cell death. This is this is something the host is trying to do. It's trying to kill itself. That way the virus can't replicate and move on to other cells. So to anthropomorphize it, it's a sort of altruistic program where one cell will take the sacrifice so that the other ones don't become infected. It's low in inflammation just by nature of it. For details I won't go into, but there's not very many inflammatory signals resulting from it. Another similar process that can occur is something called autophagy. And this is where the cell begins to eat and digest and recycle some of its components. That way it can traffic, it can have the energy it needs, and it can reduce some of these areas that viruses can get into, and it can traffic them more effectively into vacuoles and lysosomes that it can degrade viruses in. It's sort of a way for the cell to stall having to kill itself. Upon infection and recognition, cells can release cytokines. This is a primary constituent of an infection. So cytokines are these soluble proteins. You should know what a protein is if you watch the other videos, but these are, they do the work of the cell. And in this case, they are doing the work of the immune system. On the right here, in this, again, complicated crosstalk between different cell types, these are all cells of the immune system, different types. If you've watched the cell video I put up, you've recognized some of these, like the macrophage. This is a dendritic cell, essentially, or I'm sorry, this is a macrophage as well, just in a different tissue of the body. But anyhow, these cytokines right here, all of these numbered IL and TGF and TNF. Some of these are probably familiar. TNF alpha is a common one. If you have any autoimmune disease, you might have heard that. So at any rate, though, these are the way the cells will signal to each other that there's an issue, there's a problem going on, and these give very specific and direct messages to these other cell types, these cytokines. And chemokines are sort of the partner to that. Chemokines allow for basically a way to traffic cells to where they need to be. They're almost like a beacon, and they tell cells that, I have this chemokine out, and if you have a receptor for that chemokine, then you should come this way. And it helps them to get these really important T helper cells and B cells into the site of the infection. So this is a primary response by a cell which has been infected to send out chemokines. And then two other ones here. I think these are pretty neat. Epigenetic silencing through methylation and acetylation. So if you've watched the primer videos I set up, because knowing that you would need to know some of that stuff, so we can talk about it in the level of detail I want to here. epigenetic silencing works at the level of the DNA and at the chromosome level, and so, or chromatin, I should say. So one of the videos I had posted where you had, I showed where DNA was depicted as chromosomes, but it definitely doesn't always exist like that packed chromosome, and it can be in sort of this bound up, beaded shape, and that's what we call chromatin. And for a gene to be expressed, it needs to be accessible. You need to be able to open that up. And this is how the cell does it normally in the absence of infection. Methylation, just as a general rule, will tend to silence an area of DNA. That way it cannot be expressed. And this is due to some of the chemical and physical or physics properties of DNA, which I, again, brought up in those primer videos. This has to do with the charges of the DNA and the state of the DNA. So under normal circumstances, a cell can silence through methylation, and it can relax and express genes through acetylation. So a common strategy for a cell then that is infected and it has a virus which wants to begin expressing genes, it can do this silencing effect on the viral genomes, not necessarily on itself, but it can actually silence viruses that come in by methylation. I'm sure that some maybe get acetylated too. I don't know what the benefit of that is offhand, but I can maybe conjure up a scenario where it might be deleterious to a virus to acetylate it and cause overexpression of some proteins that it doesn't want expressed yet. But at any rate, that's another strategy of cells. And last here, which I think I'm actually going to do a whole video on, maybe a 5 to 10 minute video, which is this RNAi. So this is RNA interference. This is working at the level of transcription where you have that transcript. Again, I've covered in the primer videos what transcription and mRNA is. So you have that mRNA transcript, and the host will synthesize these microRNAs and these small interfering RNAs that are complementary to that viral mRNA. And so what they do is they bind it up, and now the host translation machinery can no longer make proteins, which the virus can't replicate if it can't make its proteins. And so that's I think that's a really cool mechanism. It basically sends out a complementary strand to the mRNA of the virus and shuts it down so that it can't make protein. Really neat that has evolved that way. And it's just elegant in how simple it is. But there are some steps, and just for my own purposes, I'd like to be able to describe that process, which I fully admit I can't quite remember each step, but I would like to. So maybe I'll put that up, and for people interested in seeing that level of detail, you can. So I think that's where I'll leave it for this video. In the next video, I'm going to talk about what occurs next then in the innate immune response. So this is we're still in the early stages of an infection. And at this point, cells have been infected, they've detected the virus, they've begun signaling within themselves and then externally to other cells. So what happens next? How do we get from one cell making these cytokines to a full-blown immune response? And so I'll begin to cover that next in the next video. All right, sorry this is lengthy, but it's kind of in depth. So thanks for watching.